variety can make the plant immune towards diseases? The answer is yes. That's the main purpose we trying to develop new hybrids. Apart from the yield, we also try to look at several other traits such as um, resistance against diseases, you know, or simply because you want to make the plants shorter, or you want to make the plants has this characteristic of small small fruits, or you want it to be fruits with a very bright color. So it all depends on you. But these traits need to be looked into um, in the inbred lines, in the inbred lines that we selected. So, in fact, it's not only for diseases, it depends on the traits that we want to develop. It's just that you have to search the lines for resistant or has low susceptibility against the disease. And from there, you start developing the breeding program. Examples of hybrids that are produced against diseases, you can search them in Google, lots of them.
Assalamualaikum warahmatullahi wabarakatuh. And today I'm going to present to you the learning outcomes of our SCL project. So upon completion of this part, we should be able to first describe the stage of eudicot development. And the second learning outcomes is compare the main parts of a monocot seed to those of a eudicot seed. And the last learning outcome is characteristic production of good seed. So next, seed development. So as we know, plant growth and development involves cell division, cell elongation, and differentiation of cells into tissues, which from these three, then it develops into organs of plant. So development is a program series of stage from a differentiation or specialization of structure and its function that occurs as development proceeds. So we go to table of contents. The first content is stage of eudicot development, which consists of 4 stages, which is zygote and pro-embryo stage, globular stage, the head stage, and the mature embryo. The next one is monocot versus eudicot seeds, difference between monocot and eudicot seeds. The third one is Q&A session, interview session with the specialist, which is Mr. Najib. And the fourth one is characteristic production of good seed, characteristic and example of good seeds. And 5 is conclusion of our project.
Assalamualaikum and Selamat Sejahtera. My name is Nek Alicia, and we're going to continue on with stages of eudicot development, which is a seed development is the process by which seeds are formed from end of fertilization to produce a mature seed body. So there are 4 stages. Actually, there's more, but we shorten it to 4 stages. There's zygote and proembryo stages, globular stage, heart stage, torpedo stage, and the mature embryo. So first, we have the zygote stage, which is the zygote and the endosperm become visible after double fertilization. Then the zygote will divide repeatedly until it's called a proembryo. Then suspenders transfer and produce nutrients from the endosperm. A suspender is this purple thing we have here. See, it's suspender. Okay. So next, in the globular stage, the proembryo is largely just a ball of cells, but the embryonic stage cells near the suspenders will become roots, will go to the roots and become roots. Then the cell divides with their cell plate perpendicular to the surface. Then there is a heart stage and a torpedo stage. The heart stage is the stage where it looks like a heart. The cotyledons appear and it has a heart shape. So then it will continue to enlarge and become somewhat of a torpedo shape, and the root and shoot apical meristems are going to be distinguishable in this stage. Then for the fourth stage is the mature embryo stage, which is the epicotyl contributes to shoot development. The plumule is found on the tip of the epicotyl. The procambium is located at the core of the embryo, and the integuments of this ovule become the seed coat. So this is the zygote stage, zygote and the sperm. This is for embryotic stage, dispenser. This is the globular stage and the sperm. Here is the heart-shaped stage, and this is the torpedo stage, looks like a torpedo, and finally the mature embryo stage. So that's all from me. Thank you.
Assalamualaikum and hello to everyone. My name is Siti Nurajwa binti Zulkarnain from Group 2 and my metric number is AS12280. So now, I would like to continue the presentation from my friend about monocot versus eudicot seeds. As you can see, seed development is the process by which seeds are formed from the end of fertilization to the production of a mature seed body. For the next slides, we can see that these are the difference between monocot and eudicot seeds. As you can see, the dicot seed has a shape of kidney, a human kidney, while the monocot has a square shape. So from this, we can conclude that they also have different physical shape. Only monocot has endosperm, not in dicot, but both of them has epicotyl, hypocotyl, radicle, cotyledon, seed coat, and the embryo outside the seed. But cotyledon for dicot has 2, while embryo only 1. Cotyledon for dicot usually store all the nutrients molecules the embryo uses. Meanwhile, for monocot, it absorbs other nutrient molecules from the endosperm and passes them to the embryo uses. From the previous video for interview, you can see that we provide a question about good seed, how to develop good seed. So from there, we conclude that characteristic of good seed from different variety has 4, which are physical attributes, physiological attributes, genetic attributes, and storability. For physical attribute, we wanted the plant has minimum disease and minimum damage from the outside environment, so it immune toward the disease. For physiological attribute, we have germination percentage of viability and seed vigo. Germination percentage of viability is about the seed's ability to emerge from the soil to produce a plant in the field under normal condition, while for seed vigo is about seed to emerge from the soil and survive under potential stressful field and to grow rapidly under favorable condition. So it means that from physiological attribute that we wanted the plant to survive under the certain condition. For genetic attribute, we wanted to adapt it to local condition and high yielding activity. And the last one is storability. Storability, if we have a good seed and we can keep it for a long time, so it means that the seed is a good and has a quality. So this is the example of production of good seed from different variety. As you can see, this is a palm oil palm. This is Dura piscifera. As Dura has large kernel, thin mesocarp, and thick shell, while the piscifera has small kernel, thick mesocarp, thick shell. So from both of these variety, we can combine them to make a better seed, which is Tenera. Tenera has large kernel, large mesocarp, and thin shell. So from this, the farmer can gain and obtain a lot of oil from the oil palm fruit. The last slide is conclusion. As you can see, we can conclude that if we wanted to make a good and high quality plant, we need to use clean and good quality seed. So for the next one, if we want to make a good plant, we need to choose plant from different variety, and this is also important as nowadays technologies are important to enhance the quality of plant productions. The quality of good seed produce a high yield and cost, because when the food from high quality plant, it produce a high and quality crops, the food. So people will buy the healthy food that looks beautiful. Next, production of good seed prevent and decrease disease. High quality of plant production decrease the chance of plant from getting disease. And the last one, the crops and the yield will be more healthy. So if the crops and the yield will be more healthy, people would like to buy more the food or vegetable that farmers harvest. So that is all for me. Thank you.